The Army Public Health Center conducts pooled surveillance testing for SARS-CoV-2, the agent of COVID-19, also known as the novel coronavirus. Units coordinate with APHC to have the samples pooled and analyzed by the APHC Molecular Biology Group COVID Laboratory Operations. Samples are collected from individuals at their installation, medical treatment facility, or other designated location. Samples are nasopharyngeal or NP swabs, stored in 3 mL of viral transport media. APHC is currently exploring other acceptable sampling options, such as using saliva instead of NP swabs. APHC has developed a sample submission guide for the units that provides detailed instructions on packaging and shipping the samples. APHC also provides a COVID-19 surveillance submission spreadsheet, which units must use to submit their information to the APHC lab. The installation staff log sample information into the APHC COVID-19 surveillance submission spreadsheet and properly package and ship the samples via commercial carriers to the APHC lab at Aberdeen Proving Ground, Maryland. To ensure a smooth process, it requires a great deal of coordination between the unit staff and the APHC lab. Details regarding shipment, sample numbers, and emailing of all associated paperwork have a direct impact on how many samples the lab can handle in a day. The APHC COVID surveillance sample submission form has the ability to directly import the sample information into the Enterprise Laboratory Information Management System, or ELIMS. This makes the process efficient, so by the time the samples are received, APHC will have already identified the size of the pools, pre-logged the samples into the ELIMS, printed the necessary labels, and prepared the laboratory for the day's work. As soon as the samples are delivered by the commercial carrier, the APHC COVID-19 lab team swings immediately into action. The samples arrive refrigerated and on ice in insulated shipping boxes that are suitable to ship via commercial carrier. The boxes are wiped with bleach before opened, and the packing slips are extracted to verify the samples that have arrived are the ones that were expected. The samples are immediately placed into a Class II biosafety cabinet for decontamination of the exterior plastic bags with a bleach solution. The samples are inspected to ensure that nothing leaked or was damaged in the shipping. If any samples are damaged or leaking, this is noted, and the sample is placed in an additional plastic bag to ensure no potentially infectious material leaks out. The samples are then labeled with the pool identification number and are set aside in a secondary container. Samples are then transferred to the analysts to be processed into a pooled sample. Sample receipt, decontamination, and login takes approximately 1 hour, assuming samples were prepared and shipped according to APHC guidance. The pooling process is performed in a Class II biosafety cabinet by an analyst wearing a disposable lab coat, gloves, and eye protection. A pool is made up of 10 individual samples. Samples are vortexed, and then 100 microliters of each individual sample is taken and combined in a single tube to create a pool with a total volume of 1 ml. If a pool has less than 10 individual samples, viral transport media is used to make up the balance of the volume so that consistency is maintained regardless of the pool size. Usually, one analyst working in one biosafety cabinet can process about 200 samples, creating 20 pools in about 2 hours. Pooling is by far the most time and labor-intensive part of the process. The limiting factor for how many samples can be pooled by a team of analysts pooling is the availability of both biosafety cabinets and analysts. This step cannot be automated and relies on manual labor for completion. As pooling is completed, analysts prepare plates for RNA extraction. To extract RNA from the pooled samples, APHC is currently using a magnetic bead-based automated nucleic acid extraction system called a MagMax. Each automated MagMax run takes about 30 minutes to complete and produces a plate containing up to 94 pools. That's up to 940 samples on one plate, as well as a positive control and a negative control. Before a run can be started, plates containing wash buffers, magnetic beads, binding solution, and elution buffer must be prepared. The pooled samples are added to a sample plate along with magnetic beads and a binding solution that renders the samples non-infectious. At this point, These samples can safely be worked with outside of a biosafety cabinet and can be moved onto the MagMax instrument. The magnetic beads bind to the RNA in the pools, and the MagMax moves the RNA through different wash steps until it is finally eluted into a buffer solution. At the end of the extraction process, each well on the plate holds 80 microliters of solution containing RNA from the pooled samples. These eluted samples are non-hazardous and can safely be worked with outside of a biosafety cabinet. We are now ready to move to testing these pooled samples using a research-use-only version of the real-time PCR assay designed by scientists at the CDC. To account for the dilution effect of pooling, we reconcentrated the samples by eluting in a small volume during RNA extraction. We also used a greater volume of RNA in the PCR reaction to get the same levels of sensitivity as the CDC assay reports for individual patients. The CDC assay we are using tests for 3 genetic targets. 2 of these targets are specific for the nucleocapsid or N gene in SARS-CoV-2. These are our N1 and N2 targets. These targets are specific to detect the presence of the agent of COVID-19 in a sample. We also test for the RNase-P gene, specific to human cells. This is our RP target. We include this as a control to make sure there are human cells in every sample. Because there are 3 targets, each pool needs to be tested 3 times, 1 for each target. This means that if we extract RNA from 20 pools, we still need to run 60 PCR tests to make sure that we test for everything we need. This is not including positive controls, negative controls, and extraction controls, which are run alongside our samples. 3 different PCR master mixes are prepared using N1, N2, and RP primers and light-sensitive probes, and then samples are added and the PCR tests are ready to be loaded onto the real-time PCR machine. APHC uses an ABI 7500 Fast and an ABI 7500 DX. We follow cycling conditions that are recommended by the CDC. At the end of the PCR, each of our wells will show a numerical value indicating the amount of fluorescence observed in each well. Exponential fluorescence is a good indication that the probes from each master mix detected the genetic target they were specific for and were positive for that target. We use a crossing threshold, or CT value, of 40 cycles as our cutoff to determine whether a sample was positive or if no target was detected. Positive pools are positive for N1 and N2, indicating that the virus was present in 1 or more samples in that pool. Assuming exponential amplification, the lower the CT value, the more virus was present in the sample. Inconclusive pools are positive for RP and N1 or N2, but not both. This indicates low amounts of the virus were present in 1 or more samples in the pool. Invalid pools are negative for RP. No human tissue was detected in this pool, so there may have been a collection issue with the samples in this pool. Negative pools, where no target was detected, should be positive for RP to verify that human tissue was present in the sample. Units should be prepared for any individuals in positive, invalid, or inconclusive pools to go to a local clinic and get an individual test done before they can participate in unit activities. Individuals in negative pools had no virus present at the time the sample was taken. Once the results have been confirmed for each pool, the final step is data review and then releasing the report to the submitting unit. Data is reviewed as it becomes available, and staff also conduct a formal data review in the ELIMS for quality control and assurance purposes. After this rigorous APHC review, the report is generated and released to the units and points of contact. Currently, APHC is releasing reports manually to units. Generally, reports are available in 24 hours or less after initial sample receipt, but it may take up to 48 hours or longer depending on the number of samples, how they were packaged, and other factors. APHC currently holds all samples at -80 degrees Celsius for 3 days after the report has been released in case of any necessary retesting. After that time, all samples are properly disposed of as biohazardous waste.